All right, good morning and welcome to biology for today. Today's lesson or topic of the day is going to be on bioluminescence. Honestly, a really cool part of biology, probably one of my favorite parts of biology, just simply because the view or looking at it. Just look at the background of this slide. Just think about the bioluminescence that's going on here. The glow, super cool. Always one of those fascinating things when you get to see it in person. So, got two objectives for today's lesson. First is going to be a defining objective, and then we'll have a describing objective as well. Some other small ones might be mentioned throughout. But first, define bioluminescence and differentiate it from other types of light emission, such as fluorescence and phosphorescence. And then describe the ecological roles of bioluminescence, such as camouflage, predation, mating, and communication. We'll talk a little bit more about that as the lesson rolls along. Okay, so what is bioluminescence? I ask you that question. What in your mind is bioluminescence? Well, for me, listening to your guys' answers, for me, I think of it as just a simple definition of light that gives off, or life that gives off light. And if we were in class, we'd be watching the video down below. What science says is that bioluminescence is the emission of light by living organisms through the result of chemical reactions. So another wing of science making its way in is biology. Oftentimes works hand in hand with chemistry. Okay, so animal kingdoms containing bioluminescent properties. So we have three main kingdoms, and that is protist, fungi, and animalia, so our animal kingdom. So to really understand all the bioluminescence and the groups and families that we're going to be mentioning throughout this lesson or throughout this unit, it's kind of important to touch on kingdoms and kind of talk about some biological organization. All right, so to review some classifications, or if some of you don't already know, let's talk about some new stuff. Review the classifications of taxonomy in biology. So you see this nice diagram over on the left side. We'll talk about that here on the next slide, but it'll show your organization. And we're going to start at the very, very top. So very general, very broad, contains a lot of different things, a lot of different organisms in the very, very top of this. But as you go down, it gets very specific. So let's start at the top with kingdom. Kingdom being at the very top. No, I'm not talking about castles and knights in shining armor. We're talking about animal kingdoms here. It's our highest taxonomic rank used to classify living organisms. So Animalia, Plantae, Protist, fungi is in there. So those are our three main focus points for our kingdom. Then you jump down a level. Here we are at phylum. It's the rank below kingdom, as you know. Organisms that are grouped together based on fundamental characteristics that they share and evolutionary relationships. So our two examples that are given to us are arthropods and mollusca. And you're probably wondering, okay, Mr. Stray, what in the world are these two things? Well, arthropods are, you can think of an example. Might not be able to come up with one for arthropods, but maybe mollusca. So maybe be thinking about that. As I mentioned, arthropods. Arthropods are smaller organisms. Do have some fossils of some that I would definitely bring out when I'm doing this in front of class. So definitely really cool to talk about that. They're super, super small. But you go over to mollusca, they're not much bigger, but it depends on obviously how much the organism's eating, age perhaps. So couple of different factors that can go into that. But mollusca, some of you got to be familiar with this. So think about sea life. What's coming to mind is probably maybe snails, maybe squid, octopus. That would be in the mollusca phylum. Those would be some of your examples. Then we jump down to class. So class is a group consisting of organisms sharing common attributes. So that would be aves, as it is, that's kind of for short, is the avians. That would be our birds, so ostriches. There's some other examples out there for that. Then you got reptiles, so reptilia. Can think of many, many different examples for that. I'll let you do the example finding, but aves, reptilia, that's really some of the examples for our class organization. So to review, we got kingdom, phylum, and class. So we're down, going to be going on to the order, which is the middle of that pyramid over on the other side. So here's the other four that we are going to talk about: order, family, genus, and species. Our order is a group of related families, kind of further maps out the evolutionary relationship. So that'd be the carnivora, the primates, any of those. Then you jump down another level. You're down to family, so a collection of one or more genera that share common attributes. So the Canidae, Felidae, Hominidae, so hominids. I know I probably butchered one of those saying that. But hominids, Felidae, and Canidae are three families that come to my mind when I think about the phylogenetic, or not the phylogenetic tree, but the taxonomy classifications. Genus, this is where we're starting to get very, very specific. One of our last two levels that we're going to talk about. So genus, the genus is the name. The name is the first part of it. And you see an example down there. It's always capitalized when written. So Homo sapiens, that would be us as humans. Very specific naming. I'm not going to really go into it. There's a lot of rules for the names, and there's a lot of ways that you have to write it in specific when you're addressing it in, say, you're doing a biological paper later on, maybe even in college, talking about a very specific genus. You want to make sure that you have it written properly. Otherwise, people might question what specifically you're talking about. So a lot of things go into the naming on the genus side of things, but we're going to kind of stay out of that rabbit hole for today. Species. We all know what a species is: group of organisms that share genetic heritage that are able to interbreed and create fertile offspring. So you can think of a couple: ferns, fish. The one that probably none of you thought about, and even when I created this, I didn't think about until doing some research, is viruses. Viruses are one that kind of escapes your mind because you're like, okay, well, yeah, we know they're like existing living things, but where do they fall in all this? But they fall in the species. There's many different species of viruses, many that can interbreed, change, whatever the case may be. So super cool. Don't have time to talk about viruses today, but there's a whole unit we can dive in on at a later time that talks about viruses. Okay, turn our attention back over to the left side, over to our graphic. You're probably wondering, okay, how in the world can I remember the organization here? Well, they provide a very, very nice saying that will help us remember the organization over on the left. So King standing for kingdom, Philip, phylum, can for class, only for order, find for family, green for genus, and socks for species. So King Philip can only find green socks. It's always great to have those mnemonics or, certain set of letters or different sayings in order to remember different things. So in biology, that can be very, very helpful when you have a mnemonic or a saying that allows you to remember the organization of whatever it is that you are looking at in the realm of biology. All right, so let's get a little bit more into what this lesson is about. Again, bioluminescence. Just wanted to go through some of that main information so you can kind of understand a little bit more in depth about some of the things that may be mentioned here. So first animal or first organism that you probably think of when you think about bioluminescence comes out in the summertime here in North America. That would be our North American firefly. We have a couple of these out here in western Nebraska. You don't see them very often. But if you travel back east, anywhere in Lincoln, I know for sure in Lincoln I've seen fireflies in the past. Again, one of those very common examples of bioluminescence. As you know, their flashes are very fast. 0.3 of a second in order for just one flash. And then, when you're out there looking for them, you're like, okay, where'd they go? They disappear for a few seconds and then they flash again. It takes 5.5 seconds for a North American firefly to flash. 5.5 seconds. And the light that they emit is bright enough that it has a decent temperature to it. 77 degrees Fahrenheit or 25 degrees Celsius is the temperature at which that light is being emitted. Most times, our female fireflies will wait in the grass to see if they can find a compatible male to flash with their bioluminescence. And that's kind of a way that they attract mates. There's a couple of other factors that go into our bioluminescence, but for fireflies, that is one of their reasons that they adapted to having either their certain flash pattern, their certain color, things of that nature. Again, response flash after an interval of about 2 seconds. So they can flash a little bit faster than 5.5 seconds, but I think that's more so on the side of communication. If you see a compatible mate, they're able to flash once they recognize it, and boom, that's how they're able to find mates. Again, I mentioned something about different colors. Sometimes they'll flash green, they'll flash blue, yellow. That's also how some of these species can find their compatible mates and know, okay, hey, this is a potential mate or whatever the situation may be. All right, so enough on that side of things. All right, in the back of my classroom, there are some student whiteboards. So we'll be grabbing those and doing this next activity. I like to have some activities within my lessons, kind of keep people engaged. I know that sometimes droning on and on about topics can get a little overwhelming at times. So taking a little break and stepping back and maybe doing some reviewing or some thought-provoking things could definitely keep you engaged. So with this activity, I'm not going to go through this activity with you guys here on screen, but I would just be having my students move or write on their whiteboards. They'd have 2 spots, so exist and don't exist. They're going to tell me which 3 of these organisms are bioluminescent and exist, and which 3 actually do not exist. I think it was a really cool activity. I actually learned a lot with these organisms. So definitely interesting to think about. So this activity would probably take no more 2-3 minutes unless there's some questions in class. So definitely a good good one to go through talking about bioluminescence. All right, a little more on the emission of light before we get into our fun activity for today. We're almost done with the PowerPoint and note side of things. So how are these light emissions really even possible? Well, we have to think chemistry for that and really look at it from a chemical standpoint. On the chemical side, this is a oxidation reaction. So there is this chemical, luciferin, that needs to be oxidized in order to emit this light. So if there was no luciferin, there would have to be some sort of substitute if we were going to have our bioluminescence still. But luciferin is the main chemical that is responsible for helping these organisms have this light emission. Again, oxidation, very important. Otherwise, this reaction wouldn't work, wouldn't be able to see the light. And that. Enzymes, very important when we're talking about chemical reactions in the body of organisms. You see that it's luciferase enzymes. These are the enzymes that are catalyst, and a catalyst in a chemical reaction kind of speeds up the reaction, or could just better help in bringing the chemicals that are needed to fire off that luciferin or help oxidize the luciferin that is in the system of these organisms. All right, so the main activity of this lesson deals with sea fireflies. Coolest thing that I've found in the time that I student taught. This was a fun activity I did with my 7th graders, and they actually really enjoyed it. So with sea fireflies, they were discovered back in the 18th century, very first discovered by the French naturalist Gaudet de Reville. Not sure if I got his name right, but worth the try. They were found in the Indian Ocean, so not close to North America. Indian Ocean over on the eastern side of the world. Depending on what direction you want to go, you can go to the west too. But Indian Ocean, that's where they first discovered, 18th century, not around the Americas. Then comes James Morin in 1980, who found the sea fireflies, very similar to the one pictured on the left, near the Virgin Islands. So really interesting that maybe they were by the Virgin Islands all along, but nobody had been down there to discover these organisms. And as you can tell, they're super tiny. So the only way you're going to see them is when they emit that light. But there's only certain conditions in which that can happen. Also, another cool thing before we get to the question for this slide is they're named ostracods. So a lot of times when you think of organisms in biology, they might have a nickname. So sea fireflies is the easier way, the easy name, but the more scientific term for the name of sea fireflies is ostracods. All right, so a good question for you guys to think about is, do you think it is their body that glows, or is it some chemical that they release from their body in order to have this sort of bioluminescence? Well, as we know, there are some organisms that do release these chemicals, and there are some organisms that you can kind of see this reaction going on within them because it is a transparent area on their bodies. Or for sea fireflies, for example, in this picture over on the left, their whole body is really transparent. So whenever the right conditions occur, you can see that glow. We'll be making those conditions today in our lab activity here in just a moment. All right, more on sea fireflies. I know you guys can go through and read all this left side poster. I'll put this up on the website for you guys to take a look at on your own time after class. So some other cool fun facts. 197 million years ago, this light emittance was used for defense for sea fireflies. So think of probably all the adaptations, all the reasons for bioluminescence with organisms, but we've covered two so far. One for communication with mates, and one for defense. As you can see, a few years down the road, 151 million years ago approximately, this was evolved to mating. So still used today for both. Definitely an interesting way of communication between organisms, very unique to these specific organisms. So very cool stuff. Seeing the evolution over the couple hundred million or a couple million years. What about approximately 40 million year difference. Then the last little bit. Genome research might allow researchers to track down the enzymes, the specific enzymes that are producing luciferin within these organisms. So again, there's still people out there researching these organisms, as in sea fireflies, and trying to learn a little bit more about bioluminescence. Because ultimately, bioluminescence isn't fully understood with certain organisms, so it would be really interesting to see what they discover with their genome research and what enzymes are actually helping produce the luciferin in many of these different organisms. All right, last question for today with the lesson before we get into the lab activity. So, how would bioluminescence of sea fireflies be an example of adaptation? I gave you two adaptations and a potential advantage that could such traits bring them. So again, you can use your whiteboard, talk to your neighbor. I'll call on somebody to give me their thoughts on this question, and then we will move on to our activity. So now that we've kind of taken a look at sea fireflies, let's look at what you're going to be doing on your plates in front of you here in class today. So this little vial that you see here has what we call our sea fireflies, or our ostracrods that we mentioned in the PowerPoint. These little guys, they have a really cool trick to them. So dump them out onto your plate from your vial like I did here. They're already out there. Next thing you'll need to do is get a little bit of water and get it in your pipette. And you don't need a lot of water, and just put it on your sea fireflies that are spread out on your plate. After you've done that, or even before so, kind of doesn't really matter with the order, you can crush them up with the backside of your plastic spoon, and then once you've done that and you've mixed the water with it, stir it around and observe some cool bioluminescence. Alright, so this is towards the end of the lesson, so we would be doing our hands-on activity with our sea fireflies. I'd like to have a culminating experience at the very end of my lesson, so an exit ticket might be used not every day, but for this lesson in particular, I felt the exit ticket fit a lot more. And it's a very simple one because we're going to be doing lab cleanup and everything, putting things away from our lab activity. Even though it's not a lot of materials, it does take some time for everybody to get things cleaned up and ready for the next class. So short little exit ticket for this particular lesson, which is upon completion of the experiment inquiry, Activity, please write 1 to 2 things you learned or found interesting within the bioluminescent lesson. So just a culminating experience overall from the content that was covered within this lesson.